Hello, my name is Sayaga, and today I will be introducing a suite of SARS-CoV-2 proteins that we have developed here at InBio Biotechnologies. Next slide, please. Here we are, almost 2 years in the global pandemic. There has been more than 244 million confirmed COVID-19 cases globally, with almost 5 million deaths. In the United States alone, we have seen almost 46 million confirmed COVID-19 cases, with more than 756,000 fatalities. And we are still being daily challenged, and unfortunately, it looks like that this virus is here to stay with us for some time to come. Next slide, please. Let's take a closer look at the SARS-CoV-2 virus. I'm pretty sure you've all seen different representations of this virus with its infamous spike sticking out of it. The viral genome encodes for a multitude of proteins, 4 of which are structural proteins. These include the envelope or E protein, the membrane or M protein. The E and M protein together form the viral envelope. The nucleocapsid or N protein that binds to and protects the viral RNA. And the spike or S protein that binds to human angiotensin converting enzyme 2 or ACE2 receptors located on lung human epithelial cells. And this is what that drives the infection. Today, we're going to only discuss 2 of these structural proteins, the spike and the nucleocapsid. SARS-CoV-2 spike and nucleocapsid are highly immunogenic, and most COVID-19 patients produce antibodies to these proteins. Therefore, these proteins can serve as valuable reagents in serological testing and also in research. Next slide, please. The first purified SARS-CoV-2 protein that I'll be introducing is the full-length spike. Spike is a large homotrimeric glycoprotein. It has 2 main domains, the S1 domain, that is the point of contact between the virus and its host through the receptor-binding domain, and the S2 domain that is involved in membrane fusion and virus entry. We have generated stable hex cell lines that express the full-length spike, and we've purified it using IMAG chromatography. The InBio spike has 1195 residues, and it has a C-terminal hit tag. A sample of our purified full-length spike, as analyzed by SDS page, is displayed on this slide for your view. Next slide, please. The next purified SARS-CoV-2 protein is the spike receptor-binding domain. As I mentioned, the receptor-binding domain is the point of contact between the virus and its host. We have expressed this protein in E. coli and purified it by multi-step chromatography. The InBio spike receptor-binding domain has 222 residues, and it has a C-terminal his tag. A sample of the purified spike receptor-binding domain analyzed by SDS page is displayed on this slide for your view. Next slide, please. The next purified SARS-CoV-2 protein is the nucleocapsid RNA-binding domain. The nucleocapsid or the N protein has 2 domains, the N-terminal RNA-binding domain and a C-terminal dimerization domain. The RNA-binding domain is the domain that is highly conserved. We have expressed this protein in E. coli and purified it by multi-step chromatography. The InBio SARS-CoV-2 nucleocapsid RNA-binding domain is 128 residues, and it has a C-terminal his tag. A sample of the purified nucleocapsid RNA-binding domain analyzed by SDS page is displayed on this slide. Next slide, please. Here at InBio Biotechnologies, we take pride in our stringent quality control operations. We manufacture under ISO 9001:2015 certification, which stresses on traceability and consistency. All our purified protein products are higher than 95% pure, as assessed by STS page and mass spectrometry. The IgG reactivity of our purified SARS-CoV-2 proteins have been determined using seropositive patient sera and in ELISA assays. The concentration of these purified proteins have been determined by 3 ways of amino acid analysis, OD280 and DCA protein assay. Next slide, please. I mentioned mass spectrometry as one of our quality control steps. We're very lucky here at Biotechnologies. We own an in-house mass spectrometry facility. Mass spectrometer is an excellent tool for protein analysis. We have several uses for the mass spec here at InBio. We use the mass spec for the quality control of our new allergen or protein lots to make sure there is lot-to-lot consistency. The mass spec helps us in the R&D stages of new protein products that we have on hand, and we perform protein identification through peptide mapping experiments, and we also run intact mass analysis on our purified proteins. We also run customer samples for allergen and protein content analysis. To assess the purity of our SARS-CoV-2 proteins using mass spec, we digested them with trypsin and collected the data on our in-house Q-Exactive Orbitrap mass spectrometer. Next slide, please. The purity or the relative abundance of our SARS-CoV-2 spike receptor binding domain and the nucleocapsid RNA-Binding domain were determined to be 97% and 95% respectively. An example of a peptide mapping experiment performed on spike receptor binding domain and the nucleocapsid RNA-Binding domain are displayed on right. The colored rectangles are the peptides that actually were identified by the mass spec. And when you line up these peptides with the protein sequence, you get the sequence coverage. So the sequence coverages for a spike receptor binding domain and the nucleocapsid RNA-Binding domain were determined to be 85% and 84% respectively, which is a very good sequence coverage for a purified protein. Next slide, please. The mass spec analysis of the spike, full-length spike, was a little bit different. When we first performed the mass spec analysis on the full-length spike, we only got back a handful of peptides, and this was disappointing, but we realized that the spike is fully glycosylated, so the trypsin cannot reach in and fully digest this protein. So we enzymatically deglycosylated a sample of the spike, and we reran the peptide mapping experiment. The results of this experiment is displayed in panel B. And comparing panel A, glycosylated spike, and panel B, deglycosylated spike, we see that the mass spec has been identified a large number of peptides, and we bumped up the sequence coverage from 8% to 55%. The purity of our full-length spike was determined to be almost 99% by mass spec, and the sequence coverage was determined to be 55%. A sample of the glycosylated spike along with the deglycosylated spike as analyzed by STS page are displayed on the right, on the left. If you look at the arrows, you see a migration shift for the deglycosylated spike. And it runs at its expected molecular weight of 135 kilodaltons. Next slide, please. Next, we validated our purified SARS-CoV-2 proteins for IgG reactivity in ELISA assays. In panel A, we are validating the full-length spike using 3 different sera groups and 3 different serum dilutions of 1 to 100, 1 to 1,000, and 1 to 10,000. These 3 different serum groups are internal vaccinated individuals that are displayed using green triangles. Next group are PCR verified COVID-19 patient sera, displayed by black dots. And the third group are non-COVID, non-vaccinated individuals. It is good to see that there is the first 2 groups show very high IgG reactivity against our full-length spike. And also it's good to see that the vaccinated individuals show comparable IgG reactivity to actual COVID-19 patient sera. Next. And I would like to point out to the very low background for this assay. However, this assay cannot distinguish between vaccinated individuals and individuals that actually had the COVID disease. In panel B, we are validating our purified nucleocapsid RNA-binding domain using the same exact 3 serum groups and same serum dilutions. Interesting that in this assay, the only individuals that show IgG reactivity are actually the individuals that actually caught COVID-19. Next. And I would like to again point out to the very low background of this assay. And this assay actually can distinguish between vaccinated individuals and individuals that caught COVID-19. Next slide, please. Next, we used our assay employing the full-length spike to look at the IgG titers at different stages post-vaccination. To do this, we collected internal InBio samples 1 month and 6 months post-vaccination. Then the sera were probed for IgG reactivity against the full-length spike in the ELISA assay. The vaccinated 1 month post-vaccination sera are displayed in green triangles, and 6 months post-vaccination sera are displayed with blue dots. Next, please. And we saw a significant drop in IgG levels 6 months post-vaccination. This result was both very interesting and in line with what we've been hearing on the multimedia, hence the need for a booster shot. Next. You might be wondering about this data point, the single data point in the green triangle close to 0. This individual was vaccinated by J&J vaccine, and this is 2 weeks after the vaccination. And next, please. And interesting to see that this blue dot with the arrow is the same individual 1 month post then recovering from the COVID-19 disease. So this was also very interesting to see in the assay. Next slide, please. So the applications of our purified SARS-CoV-2 proteins include in diagnostics. They can be used to assess disease prevalence in communities. They can also be used to developing rapid tests, and also they can be good tools to identify who can be a good convalescent plasma donor. And as we saw in the previous slide, they can be used for vaccine efficacy testing, and as one of our assays can distinguish between vaccine immunity versus infection. Also, these proteins can be very valuable tools in research and development of anti-SARS-CoV-2 therapeutics. They can also be used in epitope analysis experiments. Next slide, please. This is a summary of our available SARS-CoV-2 proteins. The low-tox version of the proteins is a form of the protein that has low endotoxin levels. This form of the protein is suitable for in vivo studies or cell-based assays. Usually, the lead time for this form of the protein is 2 weeks. Next slide, please. A quick note on InBio allergen and protein production capabilities. We strive for high quality, consistency, transparency, and we always have a reliable supply on hand. We are capable of expressing proteins in multiple expression systems, including E. coli, Pichia, and mammalian cells. We are fully capable of expressing and producing custom designed proteins that are higher than 95% pure, and all our protein products are validated by our in-house mass spectrometer. Next slide, please. At the end of my talk, I would like to thank the whole Allergen group, specifically Dr. Sabina Wunschman, who oversees the group, and Kathy, who has worked very hard on some of these proteins, Lisa, that finalizes all our protein lots, and Zach, who is our mass spec specialist. A special thanks to Dr. Martin Chapman, who has made all of this possible. And thanks to Dr. Anna Pomes for data analysis.